This is a video about how a small copying error on your X chromosome in a specific gene segment could lead to a disease where you chew on your lips, fingers, toes, and shoulders. Even though it is excruciatingly painful, you just can't stop. It also leads to a series of defects from low IQ to paralysis, leading to a painful and debilitating life. In the early 1960s, two brothers, a 4-year-old and an 8-year-old, were brought to the Johns Hopkins University when they had begun to show unusual growth patterns and a strange, uncontrollable behavior. The younger of the two, let's call him AD, had extremely slow growth and paralysis of all 4 limbs. What puzzled the doctors most was a peculiar movement of his hands. Even though his arms were weak and partly paralyzed, his fingers would suddenly jerk, twist, and writhe in a slow, snake-like motion. This pattern of rapid jerky movements mixed with slow twisting is called chorioathetosis. About 5 months earlier, his parents had noticed blood in his urine for the first time. Now it had happened again. Both parents recalled having mild convulsions in childhood, but nothing significant as they grew up. The older brother CE had almost identical signs and symptoms along with evidence of cerebral palsy. But there was one feature that stood out. CE had multiple scars around his lips and a deep bite marks on his fingers. One finger, after a particularly severe bite, had to be amputated. What struck the physicians was that CE felt pain every time he bit himself, yet he couldn't stop. Restraints on his arms intended to prevent injury actually seemed to calm him, improving his mood. Both the boys had the characteristic scissoring posture of their legs, which is a result of increased muscle tone from brain involvement. It was also noted that both the brothers could not sit upright without support. At the time of the evaluation, the attending physicians were Dr. Michael Lesch and Dr. Lesch-Nyhan. Through a few tests, they discovered that the brains of the brothers had decreased in size, and both of them exhibited severe IQ disability. Finally, when they examined AD's urine under the microscope, they found uric acid crystals. Uric acid is a normal metabolic byproduct formed when our bodies break down purines, the molecules that make up DNA and RNA and energy carriers like ATP and GTP. It has a pKa of about 5.4, which means that at the blood's pH, which is approximately 7.4, most of it exists in its ionized form, which dissolves easily in plasma. Normally, a small amount of uric acid is filtered and excreted in urine every day, but the presence of visible crystals tells a very different story. When excess uric acid is formed, our kidneys try to eliminate it, but as the concentrated urine becomes more acidic, these uric acid molecules lose their charge, becoming unionized and far less soluble. Once the solution is supersaturated, the uric acid begins to precipitate as crystals. Under the microscope, these appear as needle-shaped or rhomboid crystals, and this is the exact structure the physicians observed in the patient's urine. You see, there's one common disease that is known to produce excess uric acid in the blood. It is called gout, a disease of uric acid production and breakdown, causing it to build up in the joints, causing inflammation and pain. But gout typically appears only in adulthood and is usually never associated with developmental or behavioral changes. The brothers were found to be producing 200 times more uric acid than normal patients. Hence, given the partial similarity to gout, the physicians managed to lower the boys' uric acid levels by alkalizing the urine, yet that only treated one piece of the puzzle. They still couldn't understand why the crystals had formed in the first place, and why the brothers showed such wide and unusual range of behavioral and neurological symptoms. Our body relies on two vital classes of molecules to build almost everything it needs. These are the purines and pyrimidines. Their most essential role is to form the basis of DNA and RNA. The letters A and G, which stand for adenine and guanine, are purines. The letters C and T, cytosine and thymine, are pyrimidines. In RNA, thymine is replaced by uracil, which is another pyrimidine. Although both these families are used to write the genetic code, the body builds them through two different biochemical routes. Yet both begin from a single common compound called the ribose 5-phosphate, a sugar produced when our body breaks down glucose through a specialized pathway called the pentose phosphate pathway. You see, this pathway runs parallel to glycolysis. So from the carbohydrates you eat, part of the glucose goes towards energy production or glycogen storage, while another part takes a detour entering the pentose phosphate route to build the sugars that form DNA and RNA. Here's the interesting part. Unlike pyrimidines, purines are expensive to make. Each purine ring costs about 6 molecules of ATP, a huge energy investment for a single nucleotide. And because evolution always favors efficiency, our cells found a smarter shortcut, a recycling technique known as the salvage pathway. Every day we lose hundreds of millions of cells, from immune turnover to infection to aging or toxicity. When those cells break down, they release the same purine bases that we would otherwise need to rebuild from scratch. Here's a hint to help you understand how much energy is saved. You lose approximately 50 to 70 billion cells every day. Each cell's DNA holds about 6 billion bases, roughly half of which are purines. That's approximately 3 billion purines per cell. So daily purine breakdown will be approximately 1.5 multiplied to 10 raised to 20 purine molecules. If you had to rebuild those from scratch, it would cost 9 multiplied to 10 raised to 20 ATP molecules. That's about 650 calories per day, equivalent to energy received from a single meal. Instead of wasting 650 calories a day, the body salvages these purine bases to make new nucleotides and then use this energy for other important processes. You can think of it like recycling old paper into new sheets instead of cutting down fresh trees. It's cleaner, faster, and far more economical. Now, this recycling process uses a few specialized enzymes to reassemble purines back into their active forms. One of the most important of these enzymes is HGPRT, which stands for hypoxanthine guanine phosphoribosyl transferase. Its job is to convert the breakdown products hypoxanthine and guanine back into IMP and GMP, which is a raw material that can re-enter to make the building blocks. Now, it was 3 years after the incident at Johns Hopkins. At the National Institute of Health was Dr. J. Siegmiller, a rheumatologist who also served as the chief of human biochemical genetics. While working there, he came across 2 more patients who showed the same signs and symptoms as the brothers. But since he heard about the earlier cases, he had an idea of where to begin from. Just like the physicians before him, Siegmiller used gout as a comparison, but he decided to take it one step further. He tried to treat these 2 children with a medication that was normally used for gout, and the drug was azathioprine. You see, this drug is quite clever. It mimics the purine bases and tricks the body into believing that it has enough purines. And when this happens, cells slow down the purine production, and as a result, uric acid levels drop. That's how it works in gout. But there's a catch. Azathioprine is inactive on its own. It needs to be activated by a specific enzyme in the purine pathway, which converts it into a working compound. So Siegmiller compared tissues and blood samples from his 2 patients with those from real gout patients. What he found was astonishing. In the gout patient, the drug was successfully converted and uric acid levels fell as expected. And when he added radioactive hypoxanthine, it was effectively converted into IMP just as he should. But in the 2 children, neither of these steps happened. The drug never got activated, hence hypoxanthine never became IMP. That single observation revealed the answer every physician had been looking for. The enzyme responsible for this conversion, HGPRT, was completely absent. Without HGPRT, the body could no longer recycle purines, leading to an increased buildup of uric acid from the production of purines in the normal pathway. This disease came to be known as Lesch-Nyhan disease, named after the 2 physicians who had first described it. It was also explained why only male children were affected by this disease. Lesch-Nyhan disease follows what we call an X-linked recessive inheritance pattern. This means that the defective gene lies on the X chromosome, and being recessive implies that both copies of the gene must be faulty for the disease to fully appear. Males, however, have only one X chromosome inherited from their mother and a Y chromosome from their father. So if that single X carries the defective gene, there's no backup copy to compensate, and the disease will manifest. Females, on the other hand, have 2 X chromosomes. If only one of the X chromosomes carries the mutation, the other healthy one usually makes enough enzyme to prevent the full disease. Such females are called carriers. That's why almost all males with the mutation are affected, while females are usually healthy carriers. Though in rare cases due to random X inactivation called lionization, a female may show mild symptoms. During fetal growth, there's one organ that's incredibly sensitive to even the slightest chemical insult, the brain. It begins forming as early as the 3rd week of conception, and unlike other organs, it continues to develop well into adulthood. It is also shaped by the chemicals and environment around us. You see, the brain starts out as a simple tube called the neural tube. From its walls, early neurons are formed and begin to migrate to their destined locations to form the architecture of the brain. This process is highly coordinated. Each neuron follows a kind of roadway made by specialized cells known as glial cells. These guide the neurons to reach the right place and the right layer, like a scaffolding for a growing city. But reaching there is just the beginning. Once they arrive, neurons must connect, insulate themselves, store neurotransmitters, and form precise communication links. And here's where something crucial comes in. Building DNA and RNA is actually one of the smaller jobs that purines and pyrimidines perform. Their more vital role is signaling, guiding neurons during migration and helping them grow and telling them when to connect and when to stop. Finally, the brain, though only 2% of your body's weight, consumes nearly 20% of your total energy, even when you're resting. That's because neurons are constantly active, sending signals across. To do this, it needs to maintain an electric charge difference between their inside and outside, for which they use a special pump called the sodium-potassium ATPase, which runs entirely on ATP, the energy currency of your cells. And remember, ATP itself is a purine molecule, so when purine metabolism fails, signaling and ATP production is disrupted. In children with Lesch-Nyhan disease, this effect starts during early development. That's why their brain development is so severely affected. Unlike disorders that begin after birth, in utero defects affects the structures while it is still being formed. And this is why screening during fetal development can be so important, because by the time the structure is formed, some errors can never be undone. Children with LND usually show a wide range of behavioral and emotional signs. When asked a simple question, even one they clearly know the answer to, they often feel forced to give the wrong answer. It almost looks intentional, but it isn't. It is as if an impulse inside them overrides what they actually want to say. They can become suddenly aggressive, performing acts of harm or anger, yet moments later they regret it deeply and often apologize. This combination of impulsive behavior followed by immediate remorse is very characteristic. Most children also have intellectual disability, which affects their potential to follow typical schooling. They struggle with attention, planning, and execution of tasks. In one large study, about 84% patients show the hallmark sign of the disease, which is self-mutilation. Most commonly, they bite their lips, sometimes so severely that parts of the lip are lost. Fingers may be bitten repeatedly and in some cases amputated to prevent further injury. These specific signs and symptoms come from a very specific location of damage in the brain, a very important part deep in the center. If you were to take the brain, flip it, slice it halfway and zoom in, you'd see a group of structures, a kind of relay hub. It is where connections from your motor areas, emotional centers, cognition and personality all pass through on their way to other regions. This is the basal ganglia. During early development, the lack of purine salvage affects how this area forms. And because the basal ganglia depends on neurotransmitters, especially dopamine and neural connections, in LND children, the effect of dopamine and faulty development of these connections result in these signs and symptoms. Given how difficult life is for children with LND, the question naturally comes up, is there a cure? Right now, treatment is mainly about managing symptoms. Doctors lower uric acid levels, try to relieve the severe motor problems, and support the child's behavior as best as they can. Some families have to use restraints to prevent self-injury, and in extreme cases, teeth are removed so that biting doesn't cause life-threatening damage. But the future is promising. The real cure for LND lies in fixing the faulty gene on the X chromosome, and this is where modern genetics is finally catching up. Larger research projects like the HapMap, which catalog the most important regions of our genome, give scientists a detailed map to work with. Tools like CRISPR, gene editing systems, and powerful gene interaction prediction software are moving us towards a point where HGPRT defect could actually be corrected. And here's the most important part. Screening during fetal development and early detection can make an enormous difference. Catching the mutation early gives families time to prepare, also allow for interventions long before the brain is harmed. With an incidence somewhere between 1 in 380,000 and 1 in 1 million, Lesch-Nyhan disease is extremely rare. But when it does occur, the disability-adjusted life years or DALY becomes enormous. DALY is simply the years of life lost, which is the total years a person loses because the disease shortens life, plus years lived with disability. The total years lived with reduced quality of life because of severe symptoms. For a condition that begins before birth, affects brain development, causes lifelong disability, and reduces life expectancy, the DALY is overwhelming. So the real question is this. Should rare diseases like LND receive more funding and attention than common ones, knowing that the burden they place on a single child, a single family, is far greater than the numbers alone can ever show? If we can find answers for even the rarest conditions, then we create a world where every one of us, even in a 1 in a million chance, is safer.